Kia ora everyone, and welcome tonight to our digital headlines event, Understanding Memory. Before we get into the event, I'm just going to run through some housekeeping slides just for anyone who hasn't joined us before, or anyone who has, just a reminder. So just bear with me while I share my screen. Okay, so yes, welcome to our digital event. I did want to just start off our event like I do with most events by sharing this whakatauki that sits on the wall of our head of research's desk in the office. It goes ehara, takutoa, e te toa, takitahi, engare, he toa, takitini. And that stands for my strength is not as an individual, but as a collective, which we feel is very pertinent in the research world. As you can all see, your camera and microphone have been turned off and you'll be unable to turn these on, so we can't see or hear you. But if you do want to contact us, you can use the chat or the Q&A functions through Zoom. You'll find them at the bottom of your screen. And you can just click on the chat icon to use chat, and that will open up a pop-up window, and you'll see that it's set so you can only speak to the host and the panelists. So you can use this to contact us for any technical support. We'll do the best we can to help you. If you have any questions at the end of the presentation, there will be a Q&A, so you can use the Q&A function to submit your questions. That's at the bottom of the screen alongside the chat. It will also open up a pop-up window where you can type your question and it'll be visible by everybody. However, there is a little tick box where you can opt to send anonymously if you so wish. Once it is submitted, our host will then opt to answer the question live so that everybody can hear it, or we might type a response. Please be aware we're limited on time, so we might not be able to get to all the questions. And please keep all the questions related to the topic presented, and our speakers will not be able to answer any personal medical questions that you might have. At the conclusion of the session, the Zoom will end for everyone, which can be a little bit abrupt, but the recording will be available on our website from tomorrow afternoon. So I thought I'd just do a quick introduction of our host this evening, who is our Neurological Foundation Head of Research, Dr. Sarah Schomburger. And I'll just hand over to Sarah now, who is going to introduce our speakers for us.
Welcome everyone. It's so great to have so many people joining us tonight for this really special event where we've got some amazing expertise with us tonight. We have Professor Cliff Abraham, and he's been involved in memory research for 45 years. So we're going to make sure we leave plenty of time for questions tonight because of the great expertise we have. I'm sure a lot of you will have questions. And Cliff is joined by Associate Professor Stephanie Hughes and Dr. Shane O'Lean, and they all work at the University of Otago. They're involved with the Brain Health Research Centre there, and they have some wonderful information for us tonight. And I just want to thank them for taking the time to join us and to talk to the public about this important topic of memory. So welcome everyone, and welcome Cliff, Steph and Shane.
Thank you very much, Sarah. I'm going to be starting off, so I'll just get Shane to put up our screen share. So first of all, Julia and Shane and Sarah, thank you so much for inviting us to participate in this webinar. We have immense respect for the Neurological Foundation. They're a fantastic organization, as I'm sure all the attendees know, in terms of supporting neurological research in New Zealand, throughout the country, and also supporting education about neurological disorders and what we can do about them as well. So we've been very reliant as researchers on Neurological Foundation funding for as long as it's been in existence, which was 50 years or something. So So what I want to do is talk a bit about memory, but as a team, we'll be extending that to also considerations of memory disorders as well. So I'll just ask Shane to go to the next slide, which will give you an outline of the topics that we're intending to go through. So as you can see, I'm just going to start with a bit about the memory in the brain, just very basic information. Apologies if some of you already know all this, but I thought it'd be good to just run through it. And then we'll focus in on a particular part of the brain called the hippocampus and what it does in terms of memory processing. And within the hippocampus, then we're going to look at an interesting property of it, which is that its ongoing ability to give birth to new neurons throughout life. And then, of course, we know that there are many ways to have memory problems, and Alzheimer's disease is one way, and it certainly targets the hippocampus early on, amongst other related issues. Then we'll finish with a bit of what our recent research has indicated we might be able to do about it. So I'm sure that memory is critical for all our functions, both cognitive and behavioral and so forth. It's critical for animals generally to be able to move around the environment and survive, learn from their experiences, and survive for the future. And it's also very often taken for granted because it usually works really well, and often without even thinking about it or knowing about it. But equally, having said that, we're also as individuals very sensitive to when we make a memory error, which is very rare, even during normal aging. So when we have an error, it stands out because normally we're as people, our memory is usually very good. So this slide shows you how, as psychologists, we break down different kinds of memory. I'm sure it doesn't take much to imagine that there are different types of memories, and the way we break it down very basically in terms of thinking about it psychologically is that no doubt you've heard about short-term memory and long-term memory. So short-term memory is held for very short periods of time, could be seconds to minutes, whereas long-term memory can hold it for basically last for a lifetime. And importantly is that there are processes in the brain, which we'll get to in a moment, where the short-term memories can be what we call consolidated into long-term memory for future use. Within the category of long-term memory, again, very basically, we can split into two kinds. Those kinds that you're conscious of, which we call explicit memories, and those that can develop even unconsciously, or be recalled unconsciously using what was called implicit memory. And explicit memory includes two main subtypes, and that is episodic memory, which is basically about episodes in your life, and specific information about what, where, when, and so forth. Whereas semantic information is more general knowledge, and often you're learning the episodic information first, and then it passes into your semantic memory storage as you lose the sort of when it happened and so forth, but you still retain that what in general knowledge about the world. In the case of implicit memory, I guess the best examples there is on the right, when we talk about procedural memory, just basically includes things like all your motor skill learning, riding a bike, using a pencil, complicated, sporting activities, which can be undertaken without actively thinking about them once they're well learned. So, so that's something that's called, in the general category, a procedural memory. And priming is something very specific, which we don't need to get into, but is another type. So moving to the next slide, there are many types of memories, and not surprisingly, there's many parts of the brain that are involved in storing memories with kind of specializations for different types of memory. So you can see here there's the cerebral cortex, which is the large matter around the outside of the brain, which is very important throughout the whole cortex for storing different kinds of memories, but also the deeper structures shown here, like the amygdala, the hippocampus, cerebellum, basal ganglia. Basically, the bottom line is that every part of the brain has some kind of memory storage function, and even down to the spinal cord, connections there can also be involved in storing very low level but basic kinds of memory. So the brain is a memory machine. So it's designed to store information, as I've mentioned, because that's so critical for performance. And it's not like the library where a book's in a particular place, because the information can be spread across quite widespread regions of the brain. Okay, so moving on, just talk briefly about how they're formed. And to think about how they're formed, we think about, by and large, we think about the way in which nerve cells connect with each other. So here's a diagram showing what we call as a presynaptic neuron, which contacts via its axon a postsynaptic neuron via very specialized contact zones called synapses, which I'm sure you have no doubt heard of. But it's at these synapses, the strength of communication can be altered quite a bit. So it's almost like me raising my voice and lowering my voice. You'll be easier or harder for me to hear, or for you to hear me and to understand what I'm saying. So this communication between cells looks something, very artistically like this. We follow that little bright lights going down these axons, contacting the synapses and getting the next cell to fire action potentials across those specialized junctions. Of course, the nerve cells don't actually look hardly anything like this, but you get the sense that there's this interplay of signaling between presynaptic neurons and postsynaptic neurons, and the postsynaptic neurons communicate with the next set of neurons, and so forth. Okay, so moving on, what happens when certain connections are repeatedly and persistently activated through an experience that you may have or a learning episode is that these synapses can grow in size, but also in the strength of the communication between them. We refer to this as synaptic plasticity. Now, they can also shrink and become less effective at communicating. And the two things happen in parallel. And you can see here on the right a little stretch of a nerve cell process with the little protrusions coming off, which are called spines, which are where these synapses are made. And you can see that some of the protrusions have really big fat heads, and some have very small heads and very skinny processes. And these different shapes and sizes of spines that give you that tell you that there's different sizes and shapes of synaptic contacts that learning can cause growth and retraction of these spines. Between these two processes, we find that there are networks of neurons that become bound together to represent the information that is learned during an experience. And this diagram at the bottom is just to kind of provide a diagram to show that some neurons are more highly connected with each other and some less well connected. And the idea is that as a result of learning, these certain networks become very highly connected, so that if you were trying to recall that information later on, you don't have to relive the whole experience. You only need to remember certain parts of that experience, and then it will reactivate the whole network for that experience or that memory, because they're so tightly connected within the brain, and not so tightly connected to other things. And so you can relive the experience even with what we might call partial information or recall cues that allow you to bring it back. So that's the general theory about how learning occurs and information is stored in the brain in particular sets of neurons that represent those memories. This is not the whole story, but because there are other kinds of plasticity as well, which we're about to come to, but that's the kind of the fundamentals. So moving to a particular structure in the brain called the hippocampus, which is comes from originally the Greek, meaning a mythical sea monster, which has a horse-like head and a kind of dragon-shaped body, but really is also connected with seahorses, which have that kind of shape. And an actual dissected human hippocampus is there on the left, which early anatomists thought, well, that kind of looks like a seahorse. To me, maybe a dolphin or something, but that's the origin of the name. And it lies very deep in what's called the temporal lobe of the brain. So it's not at the surface, but it's buried inside, and it's an episodic memory processor. So it processes events and episodes, the what, where, and when, as I mentioned, of events, including things like spatial memory, which of course comes up a lot when we think about memory problems of people getting lost in their spatial environments. An interesting thing about the hippocampus is that it stores this information initially, but then it also works to hand off that information to other brain regions in the cortex and so forth through this process of what we call consolidation. In the next slide, it will give you an example of what it's like for someone if that ability to consolidate information doesn't occur or can't happen. And in this case, we have a patient who had neurosurgery to remove large parts of their hippocampus and surrounding brain regions to relieve their very severe epilepsy, which was really debilitating for their life. Well, the surgery was very successful in alleviating the epilepsy, but it left this young man at the time, who then lived to a ripe old age, with this inability to consolidate episodic information. In his own words, describes how he's very self-aware, as you can see, or was, he's now passed away, that he was very self-aware of his problem, because every day is alone in itself, whatever enjoyment I've had and whatever sorrow I've had. And right now, I'm wondering, have I done or said anything amiss? You see, at this moment, everything looks clear to me, but what happened just before? That's what worries me. It's like always waking from a dream. I just don't remember. So as soon as he stopped thinking about something and started paying attention to something else, he would forget about what he was seeing or experiencing before him. So you can imagine it's quite traumatic in a way. Now that's a very severe case of consolidation deficit, but people also face this in much less severe ways. And if we go to the next slide, for example, during aging, and I'm aging myself, and you have these moments and you think, gosh, I can't remember just what happened, or I can't pull some information out of memory store and so forth. And this is just a part of the natural aging process. Our ability to effectively store and retrieve information becomes a bit impaired. Not so that we can't go around and live our daily lives, and people can still be quite successful, and they find strategies to cope with that. But people often, of course, really worry about it because they think it may be a sign of something else. And that can be a case that people can be on a decline to very severe disease like Alzheimer's disease. But there's also, half the people listening will be having normal memory aging anyway, or who knows how many or so forth. So it is something to think about. And I just wanted to finish my part with a happy note. We talked about sort of declines and relatively small declines in cognitive function during normal aging. But one thing that improves for many people, if not most, is their vocabulary. So that 15,000 is the average size of your vocabulary, how many words you know. So that's not bad going if you think about memory storage, and it can continue to improve. And that's why people often find that they get better and better at crosswords. I'm still working on that myself, to be honest. As time goes on, even as they can be quite old, because they can still use and learn new information like vocabulary to help them in these tests. So it's not all bad news. And from here, I think I now turn over to Shane for the next part of the presentation.
Yeah, hi. So now we know about normal aging, and it turns out there are some nice things about normal aging, which probably you might not have known about. And one is the fact that humans don't stop making brain cells. And this was an article in Stuff in 2019, but it's actually been known since 1998 when the first article came out, showing that in the hippocampus, this really critical region of the brain, we actually grow new brain cells. So I'm sure many of you have been told that you have to look after your brain cells because you didn't grow new ones as you aged. But in fact, after we are born, we do continue to generate new neurons. And this also occurs in humans, but it also occurs in most mammals. So we can use other mammals to study these, what we call adult-borne neurons. So that really does mean postnatally. So after you're born, these adult-borne neurons. So where are they formed? And how do they develop? And what purpose do they serve? Those were some of our early questions about these cells. And they form here in this special region of the hippocampus called the subgranular zone. And turns out we have stem cells. Just throughout our life, we have these stem cells in our hippocampus. And over the course of a day, they can divide and form what we call neural progenitor cells, or these are just a precursor to neurons. And then it takes about 4 weeks for these cells to age to a fully mature cell. And here you can see it forms all these processes to join with the synapses to the surrounding cells. So this is a mature cell. And it turns out this 4 weeks of age of these cells is really important. And these cells that are 4 weeks old, and it's more of a window of a 4 to 6 weeks old, they exhibit greater synaptic plasticity than the older, more mature cells. So they participate more in the forming of the memories that Cliff talked about with the synaptic plasticity. So this is an image from our lab of these adult-borne neurons. This is what we call the subgranular zone, right along here. And these adult-borne neurons were born in an animal, in a mouse that was 4 months old. So that's like an adolescent mouse. And then these cells, we labeled them at that critical age, so we know exactly what age they are, what their birth date is. And then with this image was taken when the cells were 10 months old. So this is well into old age for a mouse. And you can see that the cells are still really functional. They still have all their processes to form synapses, and this is their cell body. So this is just an image taken from here. So the pink cells are the ones that are the adult-borne neurons. So we have a few questions about the adult-borne neurons, and one of them was what is their purpose, right? So we know already that they show in this enhanced synaptic plasticity, but we also know that when they're ablated, so basically killed off just those cells, the animals had a reduced ability to distinguish patterns, and this is something called pattern separation, which is part of the hippocampal memory scheme. Pattern separation is the ability to distinguish between things that are remarkably similar. So hopefully you can all see that this on the left is a panther and this on the right is a cat. And so our ability to distinguish between these two is what we call pattern separation. So the adult-borne neurons are really critical for this type of memory in particular. So again, more questions. One is, can we use increased neurogenesis, the birth of these new neurons in the hippocampus, to restore memory processing or to maintain neuronal function? So in order to maintain or increase the activity of these adult-borne neurons, this will ensure that they're participating in the brain circuitry. So our research questions were just two, really, to begin with. We wanted to look at these adult-borne neurons in normal aging in the hippocampus. And do they retire as they get older? Do the cells stay active, just as active as they did when they were at that critical period at 4 weeks? Or, and then if they do retire, can we activate them so that they still can be used in the circuitry of the brain? And so our hypothesis, or our first guess, was that adult-borne cells, neurons, do retire as they get older, but we needed to test this to see if it was true. and we thought that we could activate them or bring them out of retirement. And these are the results from our first study. So if we look at the activity along here, this is the activity of these cells. And these are cells that were born in young adult rats. So would be equivalent to about a 20-year-old human. And when the cells were 4 weeks old, they had this high activity that we saw, that we predicted from the synaptic plasticity studies. But then as they aged, so when they were 12 weeks old or 35 weeks old, the cells were that age, they stayed just at the same level of activity. So they maintained their activity throughout the lifespan of this animal. So these animals here at the 35 weeks are middle-aged. So we know that the cells maintain their activity all the way from adolescence into, say, their middle age, where they're about 40 to 50 in a human age. But if the cells were born in animals that were already middle-aged, they did show this heightened activity at 4 weeks of age for the cells. But as the cells age, they showed this reduced activity. So this was back to thinking about the retiring. So it's like the cells have started to retire. They're not as active and they're not really participating in the hippocampal circuitry and the functioning of the brain. So our question was, could we activate these cells? Can we change this, this reduction in activity? And in fact, we could. We could activate these old retired neurons by exposing these rats to an enriched environment. So these are healthy rats. So we had some rats that we left in a home cage, and you can see it's a rather boring home cage. This rat is all by himself, and he's just in this cage, and he of course has food and water. I've taken off the lid so you can't see the food and water, but it's just pretty boring. But then if we took some rats and we exposed them or let them play in an enriched environment, so here you can see there are 3 rats together, and they have tunnels and structures to climb on. So they're getting exercise and they're having social activity. This actually increased the activity of those retired neurons, these adult-borne neurons. So that's all in normal, healthy aging. And it's just an interesting thing about the hippocampus that we generate these new neurons, and they're worth studying. But what happens to them in a pathological condition, so in a neurological disorder? So it turns out that adult neurogenesis, so the birth of these new neurons, the number of new neurons, drops sharply in patients with Alzheimer's disease. So this study came out in 2019. And you can see in healthy humans here, the number of newborn cells that they detected was about 30,000 on average. This is the average here. But if the patients, these are human patients, if they had Alzheimer's disease, this is increasing severity of the Alzheimer's disease. When it's the most severe, they're almost a third as many cells as the healthy humans. So there's a huge decrease in adult neurogenesis in humans in Alzheimer's disease. So our next study was how to perhaps change this. And so Stephanie Hughes is going to talk about something called gene therapy, which I'll pass off to her now.
Thanks, Shane. So my part of this story is to talk about potential treatments for Alzheimer's disease and specifically gene therapy to treat Alzheimer's. But before I do that, I thought I'd start by talking about general treatments for Alzheimer's and aging in general and how to improve memory. So the most basic thing we can do is improve our lifestyle. So eat healthy, exercise more, do the crosswords and the Wordle every morning, and just generally get out and interact with many people. There's also something called cognitive stimulation therapy, which is a little bit more technical way of improving our memory that the psychologists use. And then if we move on to Alzheimer's disease, there's several medications currently available which can improve our cognitive function. However, they only really work in the earliest and mid stages of Alzheimer's disease. More recently, some of you might have heard about beta amyloid antibodies or vaccines. And there's been some negative press on that, but more recently some positive findings. So Shane, if you move on to the next one. So beta amyloid is a pathology that you see in Alzheimer's disease. So patients accumulate what we call beta amyloid plaques, and these are thought to be toxic in Alzheimer's disease. So the idea behind beta amyloid vaccinations or antibodies is that you can use them to trick the immune system into destroying the plaques and hopefully improve memory. And as I said, some of the recent findings suggest this may be helpful. But what we're doing within our groups is using something called gene therapy. So the idea behind this is that we're using genetic medicine. So we're taking DNA and we're incorporating that into a virus, or the vehicle is the virus, which allows us to package a piece of DNA, our genetic medicine, into that viral particle, and then administer that into patients. So this may seem a little barbaric that we're injecting directly into a patient's brain. However, we're now also looking at ways of doing this into the systemic system, into a vein, rather than directly into the brain of the patient. So if we move on to the next slide. So gene therapy has actually worked in a number of cases. It's available clinically for several disorders, including two I'm showing you here. So this little girl has childhood blindness. She progressively has been going blind, and she's been given gene therapy. So it's a genetic medicine administered to her eye, which has prevented her from going blind. So this is one of the very first cases of using gene therapy. Another example is children with spinal muscular atrophy, or SMA for short. They've been given these gene vectors directly into their spinal cord, which prevents the devastating effects of this disorder, which is generally fatal within a few years. So with this treatment, these children are living so far very healthy lives. So gene therapy can work. What we want to know is, can it work in Alzheimer's disease? And as I mentioned, we really don't want to be injecting these viruses directly into the brain. We'd rather be able to do it through the systemic system or the venous system. So to do this, we've had to rederive or remake some viruses to improve their ability to go from the body into the brain through something called the blood-brain barrier. And I think now Shane is going to tell you how we've been doing that. No, before she does, I'm going to tell you about the mouse model. So I mentioned plaques already, the beta amyloid plaques in humans. We have a mouse model that we use that also accumulates these amyloid plaques. Shown here is the hippocampus. This is a hippocampus of a normal, healthy mouse. And down below that, you can see the plaques shown in red. Okay, and these are the amyloid plaques, which accumulate in the model of Alzheimer's disease. And now I'll hand over to Shane.
So hi again. So we used this gene therapy method to overexpress a protein, which we call SAPA. This is SAPA. And the first study that I'm going to talk about is we tried to increase this neurogenesis. So to reverse the trend that we saw in Alzheimer's disease, when I talked about in patients, where we saw a decrease in neurogenesis. Well, in our Alzheimer's model mice that we just talked about, we also see that decrease in neurogenesis. So if we look at, these are the healthy mice here, and these are the new cells. You can see they have this level of new cells, and then the Alzheimer's mice have a much lower number of these new adult-borne neurons. So if we use our gene therapy and treat with what we call SAPA, there's no difference in the normal healthy animals, but in the Alzheimer's disease mice, we show an increase in the neurogenesis, so the birth of the adult-borne neurons. So we've restored to the control level the adult neurogenesis. So this was done with the traditional gene therapy, but more recently, we've used a systemic administration of the gene therapy into Alzheimer's disease model mice. So what we call a peripheral administration. So it means it's not directly into the brain. And we found that we could reduce the plaques, the number of plaques in these Alzheimer's disease mouse. So this is just in the hippocampus. And you can see in a control mouse, there are no plaques. That's a healthy mouse. If we have an Alzheimer's mouse, they have a lot of plaques here. And then if we treat them with the gene therapy to overexpress SAPA, we see a much greater reduction in the plaque load. And the exciting thing about this is this was in the hippocampus, but because it was an injection into the vein, so covering the administration of the virus, it went to the whole brain, it also worked in the cortex. So again, this is that outer shell of your brain. The healthy mice have no plaques, and the Alzheimer's mice have a lot of plaques, and it's reduced with this gene therapy overexpressing SAPA. But really, the gold standard is, can we restore memory with our gene therapy? So can we restore the memory in Alzheimer's disease? So we have an experiment. This is also overexpressing SAPA. So this is an experiment where a mouse has to find a platform that they're initially shown where it is, but then they have to remember later where it is and find that hidden platform, which is a safe place for them. So if you look at a healthy animal, on the first day, they take, quite a long distance to get to that platform. But over time, they learn and remember, and so they take a shorter distance each day to get to the platform. So they have learned and remembered, so they have a healthy memory. If we look at an untreated mouse with Alzheimer's disease, they travel almost the same distance every day. So they haven't really learned and they haven't really remembered where that platform is. So it's significantly different from the healthy mouse. But if we treat them with the gene therapy with the overexpressing SAPA, we found we restored the memory. So they also learned and have a shorter distance each day to find that hidden platform. So we were able to rescue memory in this Alzheimer's model. So in summary, for our results, the first one was new neurons are born in adults. Hopefully that's taught you something tonight. If these cells are born in when we're young adults, they maintain a high activity all the way through to middle age and perhaps even longer. If the cells are born when you're middle-aged, these cells can be reactivated by an enriched environment, which in humans probably translates to a lot of social activity, exercise, and solving new puzzles and keeping cognitively active. We can increase the number of new cells in a mouse model of Alzheimer's disease when we overexpress SAPA using gene therapy. Using the same gene therapy, we can reduce the plaque burden in Alzheimer's disease, and we can rescue memory deficits by using these gene therapies to overexpress SAPA. So finally, as we heard at the beginning, it's all part of a collective effort. All of this work has been done by all these people here, and the list here, of course, you've seen Cliff and Stephanie tonight, but also Professor Warren Tate, and then the students who have been involved, Connie Chen and Valerie Tan did quite a lot of the work that I've talked about tonight, and then Barbara and Lucy are part of the original Neurogenesis team, and then so several other students, Emily and Sophie, have also worked on this gene therapy. And of course, at the end, I really want to thank the Neurological Foundation. None of this work really would have been possible without their support, and especially thank you to the donors who have contributed to the Neurological Foundation, allowing this work to go forward. So thank you very much. And we welcome any questions.
Thanks so much, Cliff and Shane and Stephanie. And if you can all come back on your cameras now. And the first thing that I'm really keen to know is, where is this in the process of potentially getting the gene therapy into humans? I might pick on you for this one, Cliff. What would be the next steps and the sort of timeline?
So that's a very good question. Thanks, Sarah. And you know, Stephanie is actually our virus expert, but I'll make a stab and then see if Stephanie wants to kick in. So we have a hurdle that we have yet to cross, and the hurdle is that the you know, this particular virus, and it's not really that it might sound scary to people that we're using a virus, but we're not really using a live virus. We're using just the virus envelope to package the particular gene that we're interested in. So it doesn't replicate in the animals, it doesn't make them sick. And unfortunately, this particular virus envelope that is being used for our studies doesn't seem to work and get across into the brain from the periphery in primates. So that means it's unlikely to work in humans. But thankfully, the Neurological Foundation has just funded a new project for our group to try to use what a new virus envelope that has just been recently reported that can cross the blood-brain barrier in primates. So if we can replicate our findings using this new virus envelope with our SAPA gene that's being packaged inside it, then that will really be the real trigger and excitement point for us that we may be onto something that could move to a clinical trial.
Well, that would be really amazing. Do you have anything further to add on that, Steph?
No, Cliff's explanation is a really good one. We have to not only go through some more mouse trials with this new virus, but then probably go to a larger animal model. So either a non-human primate, monkey, or sheep model before we'd be able to get to a clinical trial.
So one of the questions that has come from the audience is about stem cells and whether they are involved in the gene therapy. Do you want to answer this one, Steph?
Yep, sure. So in our gene therapy, we're not using stem cells. It may be that some of the viruses that we're putting into the brain are getting into those neural stem cells that Shane was talking about. But the way that stem cells are being used currently in gene therapy is with bone marrow stem cells. So many of these gene therapies, we would take the stem cells from the bone marrow of a patient. You take them out of the patient, you target your gene medicine with the viruses in a dish outside of the body, and then you'd put them back as a bone marrow transplant into the body. And that's been tried for quite a few brain diseases, as well as a lot of blood disorders like hemophilia. So it is possible to combine gene therapy with stem cells.
Sarah, I might kick in there as well. So it's a good question about stem cells, and obviously we're, you know, I think it's a great thing that this treatment that we've been developing promotes the neurogenesis, as Shane described. But the thing to be aware of is that, you know, very few parts of the brain actually undergo this type of neurogenesis as adults. So as I said at the start, memories of different kinds are stored in various places around the brain. So if we were just trying to increase the number of adult-borne neurons, it might be helpful for the hippocampus function, but it wouldn't necessarily help for other parts of the brain where there's also memory difficulties. So we need to find therapies that have wider function than just promoting the neurogenesis. And the SAPA protein does just this. It does help with the neurogenesis, but then it also has many other functions, neuroprotective functions that would help with that would help with attacking the disease mechanisms.
And are there any other groups around the world working in the same field, or are you collaborating with any others around the world at the moment to try and get this gene therapy to progress towards clinical trials? I'll put this one to you, Shane.
There are definitely many groups looking at gene therapy. I think we may be unique in the proteins we're choosing to express and the things that we are studying. directly, but gene therapy is indeed being studied by many other groups, and for other neurological diseases as well, not just Alzheimer's disease. So
one of the audience has asked a question about a program called the Brain Docs, run by Doctors Aishi and Dean Sherzai. Do you know of that program and whether programs like that are good for people to follow for their brain health? I'll put this one to you, Cliff.
I don't know off the top of my head what that program is, but look, there are many brain training, brain health programs around, and I think the ones that are likely to have the most effect are ones that have multiple elements to them, and you know, just doing crosswords isn't going to stave off Alzheimer's disease, for example, particularly if you're just staying by yourself and that's all you do. You'll become very good at doing crosswords, but that's probably not enough. So you need to have these different elements that Shane has mentioned. I don't know this particular program. I don't want to pass comment as to whether it's a good one or not. I think the bottom line message is, however you choose to do it, and there's many ways to exercise. You don't have to go out into the gym. There's, you know, gardening or going for walks or going shopping, you know, in a big mall. Using your brain and the social contact is quite important. So as well as good nutrition, of course. So yeah, you need to have really build in all those elements for, I think, maximal protection. It doesn't mean a person won't get Alzheimer's disease, but it means that it would push out much later in the onset.
So there's good evidence in research that people who are really focusing on good nutrition, exercise, socialization, and learning new things are doing better in terms of brain health?
Yeah, I think that's safe to say. You know, these are quite difficult studies to do because they take it takes a long time to look at this sort of thing. But there's a lot of longitudinal studies now where this can be broken out and can be identified, as well as the active training or active programs, you might say, that are being undertaken. So yeah, I think there's general agreement about these aspects. You know, what is the exact best kind of nutrition, and what are the exact best foods can be debated. But in general, you know, a healthy diet, lots of fruit and vegetables. You know, the less alcohol, the better. Stopping smoking. There's many things that people can do to increase their brain health.
I'd like to.
Oh, go for it, Shane.
I just wanted to say something because when we were putting this talk together, we just had a wee discussion about the fact that Alzheimer's disease is now becoming more seen as a whole body disease and not just that of the brain. I mean, it's of course, you know, the whole body contributes to the healthiness of the brain, but I think that it's borne out in what Cliff was saying in terms of, you know, exercise and social interaction. It's a whole body disease. So.
We also have a question from the audience about how stress impacts on memory. Would you like to answer this one, Cliff?
Yeah, well, there's two elements to stress, I suppose. Typically, we would break it down: acute stress and chronic stress. So acute stress generally sharpens your narrow focus, and so a lot of things that may have been happening outside that narrow focus you don't remember very well, but you can become you can develop a hyper memory. This is called post-traumatic stress disorder. So now a highly emotional or stressful event gets repeated over and over in your mind as you ruminate about it, and that can be a super strong memory, very difficult to erase. So acute stress can have both kinds of effects on memory function. But if you are under chronic stress, for whatever reason, this, for a start, impairs neurogenesis in adults. So you're losing the ability to produce those adult-borne neurons so easily. And it actually starts causing neurodegeneration in parts of the brain involved in memory, including in the cortex and the hippocampus. So yes, definitely chronic stress can have severe impacts on your memory functions. And probably not just your memory functions, but that's one that people, I mean, the question, I think indicates that people are particularly worried about that particular cognitive function, but it probably impairs other parts of your cognitive structures as well.
So Steph, we have a question about when the virus is injected into the brain. Does it get through the blood-brain barrier, or does it just get into the brain itself? How does that work?
Mm-hmm. So we've shown you two examples tonight. One of them is where we inject directly into the brain, so we bypass the blood-brain barrier. You can also do that by injecting into the spinal canal. You also bypass the blood-brain barrier. And that's been used in quite a few different gene therapy applications, including spinal muscular atrophy that I showed. Or you can use this new viral vector where you can go into the venous system systemically. And in that case, you do have to cross the blood-brain barrier with the virus. So these new viruses have developed a way of actually crossing that barrier into the brain. So both work. One's just less invasive than the other.
Someone is also asking about the different anti-amyloid treatments. I know this is a controversial area in Alzheimer's disease as to whether the amyloid is causing everything or if it's just a byproduct. Do you want to comment on the different treatments out there? You mentioned a couple of them, Steph, have had promising results recently.
Yeah, so the most recent one was last week, where some of the larger studies using the anti-amyloid vaccine basically looked like it was having some positive effects, whereas a lot of the other ones have been discontinued. So I think the jury's still out as to whether just removing the plaques is going to have an effect. And part of the reason why we say that is there are many individuals who have amyloid plaques but never show any signs of Alzheimer's disease. So if its cause or effect is really unknown at this point. But at least the more recent data looks promising. It just needs to be repeated.
I might jump in there as well. I mean, I think it's too simplistic, to say whether it's the cause or it's a byproduct. It is definitely not normal to have amyloid plaques, and amyloid plaques causes neuroinflammation, which is common in many brain disorders, from stroke to head injury to neurodegenerative diseases. The brain's immune system gets into gear and starts trying to help out. And plaques activate this immune system too. And so, plus they're space-forming lesions in the brain, so they're putting pressure on the normal tissue. So if this goes on for quite a long time, that does cause deficits in neural functioning. But whether they are the primary cause, I mean, the question is, why do the plaques form? So there's something happening beforehand that's causing the plaques to form that people still don't understand. And it may be because the blood flow to the brain's becoming impaired, or something to do with the immune system, something happening in other parts of the body, like Shane was saying, that's driving in an immune response that gets into the brain. So amyloid is definitely part of the disease. It's pretty specific to Alzheimer's disease for these particular kinds of plaques. But whether attacking specifically the plaques and only the plaques will be sufficient to fully treat the disease is the open question.
So obviously your treatment is very specific to the amyloid plaques and Alzheimer's disease, but you did mention that there is gene therapy around the world for a lot of different disorders. So I might bring this question to you, Shane, because people are asking about potential gene therapy for brain tumor recovery or stroke. Do you know what other conditions are making progress in the field of gene therapy at the moment?
I definitely wouldn't be the expert on this one. I would say that Steph would be able to speak much better about what other diseases gene therapy has been targeting. But to my knowledge, I don't think that bipolar disorder has been investigated, or tumors. I don't think. But yeah, Steph, maybe you can. Yeah,
there's a lot of preclinical studies going on for many of these disorders, and there's a lot of clinical trials now in humans for a huge range of disorders. Most of them, I'd have to say, are genetic diseases, so where you have mutations in specific genes, and what you're trying to do is put back a normal functional copy of that gene back into the brain or wherever it's required, depending on disease. So these disorders, like Alzheimer's, that generally are not one single genetic mutation, are slowly coming on board when we find these sort of genes like SAPA that can have some sort of neuroprotective role. So it is coming, and there are some tumor therapies out there that are gene therapies. But yeah, the whole field is just exploding at the moment.
So we've heard that the ApoE4 gene can make people more susceptible to Alzheimer's disease. So how would this be involved in gene therapy, or how would it affect the trials?
Yes, you're right, it's a risk factor for Alzheimer's disease. In terms of the gene therapy strategy that we've got, I don't think that SAPA is going to be more or less effective based on your ApoE4 status. But Cliff might have some comment on that too.
Yeah, I mean, there's a specific form of ApoE called the E4 form that is the particular risk factor, and that affects the handling of the amyloid that occurs naturally in the brain. You shouldn't get the idea that amyloid is bad, because it occurs normally, but in the disease state, it starts to aggregate into these clumps, and that's where it becomes more of a problem. And so part of the problem may be exacerbated by having the E form of the ApoE protein, because that affects the handling of molecules, including amyloid. So yeah, it is definitely a risk factor, but it's also a risk factor for other things like head injury and stroke outcomes and so forth. So it's it is, but it doesn't mean that if you have that gene that you're going to get Alzheimer's disease. Mm-hmm. So it's a major risk factor, but it doesn't determine that you get the disease.
So I guess Alzheimer's has a lot of factors, potentially genetic risk factors as well as environmental. Would you say one is more important than the other, or do you think there's a lot of different things going on in Alzheimer's that we're still learning about?
It's in a combination of the two. There are some rare genetic variants that cause very early onset Alzheimer's disease. But for most people with a later onset, it's a combination of genes and environment. So you might be dealt a bad hand with genetics, but like even the ApoE4, it doesn't mean that you're going to get Alzheimer's disease. It just increases your risk of developing it. So it's very much a combination of the two.
Some lucky people actually have genes that are, forms of genes that protect them against Alzheimer's disease. So in some ways, those people and those gene variants will be very interesting to study, to see how they protect against getting the disease. And while we're talking about the genetic form of the disease, which is only a few % of all cases of Alzheimer's disease, I see one of the questions is how do the mice get Alzheimer's disease? So I might just segue to that just real quick and just say, that's one of the great things about being a mouse is that you don't get Alzheimer's disease. So in order to have a mouse model, and I mean, very few animals do get Alzheimer's disease. There are a few, but for mice, you have to make what we call a transgenic mouse, where they start expressing one of these human genes that we know causes the familial or genetic form of the disease, and then they start producing this disease pathology as a result.
So in terms of mild cognitive impairment, if people have been diagnosed or they've noticed memory issues and they've changed their lifestyle, have they been able to increase neurogenesis and reverse the cognitive deficits that they've seen? I'll put this one to you, Cliff.
It's not that easy to study neurogenesis in people, particularly while they're alive. It can be kind of done, but almost all the work is done with, using brain banks where people have donated their brains for study post-mortem. And I'll just give a shout out here to the Neurological Foundation Human Brain Bank that's based in Auckland, where a lot of work is being done, tissue is being available for research, both in New Zealand and around the world for this kind of thing. But maybe I could rephrase the question: is it ever too late to start some of these lifestyle changes? And the answer is no. So it's never too late to start and try to hold at bay any future decline or further decline or push it out. So whether that involves changing neurogenesis or not, we can't say. And quite likely, as I say, we need to affect more than just the hippocampus. So these changes will be happening throughout the brain. One of the things that happens with enriched environment exposure and exercise is that that brings up the natural levels of the SAPA protein as well. So it helps engage the natural protective kind of weapons that the nervous system has to try to fight off disease and keep as healthy as much as possible.
So we've gone past our 8 p.m. time. Do you all have time for a couple more questions before we close out? There was one that came through earlier that was quite interesting about prospective memory, which is where you plan something in advance and you remember what you've planned in advance. Cliff, where does that sit in your diagram of different types of memory?
Yeah, that's probably come up in popularity in thinking about memory functions after the classical definitions that I put on the slide. But it really is a kind of episodic memory. There's some information and you need to retain it and store it for future action, usually within relatively short time frames, but I suppose you could be planning for something to happen months or a year away. But it's kind of a funny thing to call memory because the things haven't happened, but it is recalling your plans and that you've developed for future action. So they probably fall under the rubric of episodic memory because the information is being accumulated in a specific time and place as you're developing those plans.
So I think we're going to end on a question just to wrap up about Alzheimer's disease itself and how it would be diagnosed if people generally suspect they might have it, or if you have it, you wouldn't really know. Do you want to comment on that, Cliff, about how someone would go about getting diagnosed?
Sure. It's an inexact science at the moment. But the best thing to do, of course, if someone thinks that they or a family member or a friend is having a memory issue that is getting worse over time, then they should definitely see their GP, and they'll probably be referred to memory clinics who will be in the hospitals or Fataora, and they will be getting assessments as to whether they have memory impairments characteristic of Alzheimer's disease. I mean, many types of disorders and not necessarily diseases can cause memory problems. You can have memory problems because you're not sleeping well, because you're depressed, because your medications you're taking for something else are actually depressing memory. So you need to rule out all these other factors and have a proper history done in order to kind of begin to think that, okay, maybe this is Alzheimer's disease. But there are also various kinds of dementias that also have memory problems as well. So it's usually not possible to definitively say someone has Alzheimer's disease unless they are actually studied, with their brains are studied post-mortem and look at the specific pathology. So at best, you might be characterized as having probable Alzheimer's disease. But whether it's Alzheimer's disease or frontotemporal dementia or vascular dementia or other types of dementia, basically the same lifestyle interventions that you want to kind of implement in terms of the treatments that are available, they're very similar in many cases. So you just have to find ways, strategies to kind of deal with it on a day-to-day basis as best you can. I will say that we are also working here at Otago, as others are around the world, to find more and better diagnostics. So of course, brain scanning is one way to kind of up the ante in terms of knowing whether you've got an Alzheimer's disease-like changes in the brain, including reductions in the hippocampus size and shape. But those are expensive and not easily done on a widespread basis. So there's a lot of work that is looking at whether we can develop blood tests for Alzheimer's disease. And again, the Neurological Foundation has funded some of our work on this, which has been fantastic. And we've developed, a promising test that needs, further validation. But and others have also produced tests, tests and measures of different molecules. So we're gaining ground on getting better and better diagnoses based on even what can be relatively simple things like blood tests.
Yeah, I think there's some very exciting things going on in the space, and I know you all have a lot of projects going on, and we've just talked about one of them tonight. And I just want to acknowledge how successful all of you are with getting funding. It is difficult in New Zealand, and you've all got funding from some of the really major funders to do this exciting work and keep progressing it. And we just really wish you all the best with it. And we really want to thank you also for coming and taking time to talk with us tonight. If people are interested in hearing more about research tonight, we actually have our Chair of Clinical Neurology, Alan Barber, on Radio New Zealand tonight, and he'll be talking at 9 p.m. And if any of you want to stay up longer and listen to more research, then tune into Radio New Zealand tonight at 9 p.m. to hear him talk about his important work in the field of stroke. So I just want to say another big thank you to you all, and I want to say thank you to all of our audience for joining us tonight. It's been fascinating, and the talk will be going online as well. So ka kite, good night.
Ngā te wā.